Now the transport of the samples for the viruses, what are the media which are going to use for the transport of the viruses, which are the precautions should be taken for the transport of the viruses, for the collection of the viruses, for the collection of the viruses samples, and then transport towards the laboratory. Purpose of these standard operating procedures: safety from the biohazard material. Of course, we have already studied that these are the viruses, and most of them, some of them can be the zoonotic importance. The zoonotic are those diseases which can be transmitted from the animals towards the human beings and the human beings towards the animals. So therefore, these are very important. If you are going to take the samples from the animals, you need to be careful. You will see in detail. And the results depend upon the proper sample collection and proper handling. Of course, we are going to work hard, and you are going to collect the samples from far away in the field, and then you are going to send it to the laboratory in any of the, or maybe in the big city. So it is important that you need the results you are expecting for. Therefore, if you are not going to do proper handling and proper collection and proper transport, you are not using the proper transport media, or you are not handling it in the proper temperatures, so you will not be able to get the proper results which you are going to expect. First of all is the biosafety concentration is the most important. Whenever you are going to collect or transport any of the samples which are including the viruses, you need to think about its zoonotic importance and transmissible importance. Whether what kind of virus you are expecting for, whether it has the zoonotic importance or not, whether viruses can be transmitted from the, if you are going to work with the animals, then you need to be more careful that maybe you are going to act on such kind of the viruses which are also transmittable to the human beings. Therefore, you need to use the proper handling procedures. Latex gloves, lab coats, and masks are always suggested whenever you are going to collect or handle. New syringes and containers are very important. Don't use any containers which are not autoclaved or not sterilized, and similarly the syringes should be used, if they are disposable syringes you need to use, this should be the new syringes. And then the container should be sealed enough so that no virus can come out at the time of the transport, maybe because on the way of the transport maybe it is not handled well. Therefore, before sending it for transport, the container should be sealed enough. Wash hands after removing the gloves, still need to be care. And if you have some kind of the injury, it's better not to handle any viral infection, any viral samples. And if there is injury after the handling, you need to wash the area with the soap and the water. Always have the first aid safety kit with you. These were the biosafety considerations. Now handling, we can see the different kind of the samples, and then handling and the transport of the blood sample. Collect EDTA or heparin containing containers. If you are going to collect the blood samples, it is better that container should already contain the EDTA or heparin so that blood sample should not be wasted. Store at 4 degrees centigrade just after collecting. Transport upright with the cushion to prevent the hemolysis, and never freeze blood. However, these days we already have the containers which are available in the market which already coated with the heparin or EDTAs. So just take the blood and take care that you are transporting it in the upright cushion with the cushion. Therefore, it will not be wasted, and there should never be freezed. The blood samples will never be freezed to avoid the lysis of the red blood cells. Then if the samples are the nasopharyngeal swabs, then how will you are going to transport such kind of the samples? Because of course there are the respiratory signs, and the nasopharyngeal swabs you will take in case of the respiratory infections. The respiratory infections need the proper medium for transportation. The respiratory specimen should be transported in the proper medium. We have already studied the detail of the medium, that for which kind of the specimen, which kind of the media is needed. Care should be taken for the time, because such kind of the samples needed to be transported as quickly as possible to the laboratory, because there may be the overgrowth by the oral flora. For transit period up to 24 hours, it is better that they should be reached within the 24 hours. And because it may be the ambient temperature, it depends on the bacteria, and it may be avoid the contamination with the bacteria. And for the viruses, it may be 4 to 8 degrees centigrade. Then the postmortem samples. Sometimes we are going to collect different kind of the samples and we are going to use them. Sometimes the animal or the samples are the already died. So we need to take the samples from the postmortems. Biopsy relevant tissues in formalin for the histopathology. It depends on what kind of the test, which kind of the experimental test or investigation is going to act upon on such kind of. If you are going to do the histopathology, then the samples should be taken in the formalin. In transport medium for microbiological testing, if you are going to do some serological test or molecular biology tools, then it's better to use it in the proper transport medium. In sterilized line for isolation of the viral pathogens. Now the handling and the transport. The transport media within the 24 hours, as we already discussed, with the ambient temperatures and the sterile saline at 4 to 8 degrees centigrade, and it should be reached in the laboratories within 48 hours after the collection of the samples.